Hello, my name is Joshua Gabi. I welcome you to Reproduction in Living Organism, questions 41 to 45. Explain the postnatal changes in the human embryo. Part B. What is the significance of the changes mentioned above? Part C. How is the mammalian placenta adapted for the transportation role? Explain the postnatal changes in the human embryo. Circulatory postnatal changes. Systemic vesicular resistance and aortic pressure increase, raising the pressure in the left atrium and ventricle too. Pulmonary vesicular resistance greatly decreases as a result of the expansion of the lungs. Closure of the foramen oval due to the low right atrial pressure and high left atrial pressure. Functional closure of ductus arteriosus. The increased systematic resistance elevates the aortic pressure, while decreased the pulmonary resistance reduces the pulmonary arterial pressure. Closure of ductus venous, raising the portovenous pressure. Initial breathing once placental connection is cut off due to the initial hypoxic state during birth. Expansion of initially collapsed alveoli and hence expansion of the lungs. Surfactants production in the lungs by the alveolar cells. Part B. What is the significance of the changes mentioned in A above? The rise in the systemic vesicular resistance compensates for the tremendous blood loss through the placenta, preventing sudden drop in the fetal blood pressure. The decrease in the pulmonary vesicular resistance prevents the compression of the pulmonary blood vessels because of the small volume of the lungs maintaining the pulmonary blood flow. It also reduces the pulmonary arterial pressure, right atrial and right ventricular pressure. Closure of the foramen ovary cuts off the right-left shunting of blood in the heart, permitting channeling of deoxygenated blood to the lungs for the oxygenation. Functional closure of ductus arteriosus permit initial pulmonary circulation. The reason portovenous pressure. After closure of ductus forces the portovenous blood flow through the liver sinuses. Initiation of breathing once placental connection cut off ensures gaseous exchange, hence prevention of tissue hypoxia. Expansion of the initial collapsed alveoli and hence expansion of the lungs permit initiation of gaseous exchange via the lungs of the fetus. Surfactant production in the lungs by the alveolar cells prevents alveolar collapses that can otherwise result in respiratory stress syndrome. Part C. How is the mammalian placenta adapted for the transportation role? Closeness of the maternal and the fetal blood vessels facilitates faster diffusion of substances. Chorionic villi cells, they contain numerous mitochondria to provide energy required for active transport. Numerous chorionic villi, they increase the surface area for absorption and transportation of material. The two umbilical arteries transport blood containing wastes from the fetus to the mother's blood for excretion. The umbilical vein, this transports nutrients and oxygen-rich blood from the mother to the fetus. The numerous maternal arterioles, they supply oxygen, nutrient-rich to the fetus. Numerous maternal venues, they drain the fetal blood containing wastes to be transported back to the maternal blood for excretion. Chorionic arteries, they divide to cotyledon arteries, further divided to form dense arterial-capillary venous system, create a steep concentration gradient, allowing efficient material transport and exchange. Selective permeable placental barrier membrane. This allows selective diffusion of antibodies for passive. Compare the processes of the development of the pollen grain and the ovule. Part B. Describe the processes leading to the fertilization of an ovule after pollen has landed to the stigma. Compare the processes of development of the pollen grain and the ovule. Shall first look at the similarities. Both the ovule and the pollen grain, they rise from a diploid mother cells. And both mother cells divide mitotically. In both, a tetrad of haploid cells is formed from the mother cell. In both, mitotic division of the nucleus occurs. Let's look at the differences. In pollen grain, arise from the pollen mother cells. The ovule arises from the megaspore mother cells. Pollen tetrad cell formed from the mother cells develop into pollen grain. In ovule, only one of the tetrapod cells formed from the mother cells develop into an ovule. In pollen grain, nuclei of the resultant haploid cells divide once by mitosis. In the ovule, the nucleus of the remaining megaspore cell undergoes two successive mitotic division. In pollen grain, the nuclei remain at the center of the cell. In the ovule, the nuclei resulting from the division of the embryo sac nucleus migrate to opposite poles. Part B. Describe the processes leading to the fertilization of an ovule after the pollen has landed on the stigma. On landing upon the stigma, the pollen grain adheres to the stigma by a sticky substance secreted by the stigma cells, absorbs water, sugars, and germinates to give the pollen tube. Then the pollen tube grows into the stigma down the style to the ovary, pushing its way between the loosely packed cells and deriving the nourishment from the surrounding tissues. The pollen tube nucleus is positioned at the tip of the growing pollen tube, controlling the growth, while the two male nuclei, the two sperm, formed by mitosis from the generative nucleus, follow closely behind chemical substances in the style. Embryo sac enables the unidirection rapid growth of the pollen tube towards the ovary. After reaching the ovary, the pollen tube grows towards the ovule and usually enters through the micropyle, the chalaza, or integuments. Then the pollen tube reaches the center of the ovule, penetrates the wall of the embryo sac, bursts open, and the tube nucleus disintegrates. One sperm male nucleus fuses with the egg cell to form a diploid zygote, while another sperm, the male nucleus, fuses with the two polar nuclei to form a triploid primary endosperm nucleus. This is called double fertilization. Distinguish between fertilization in flowering plants and mammals. Part B. Give an account of the events which occur in the oviduct leading to fertilization. Part C. What are the advantages and the disadvantages of internal fertilization in mammals? Distinguish between fertilization in flowering plants and mammals. In flowering plants, it occurs in the embryo sac. In mammal, fertilization occurs within the ovule or the egg. In flowering plant, there is double fertilization. Two male nuclei are involved. In mammals, single fertilization. One male nucleus is involved. In plants, haploid male nuclei fuses with both haploid nucleus and diploid nucleus. In mammal, haploid male nucleus fuses with only one haploid nucleus. In plants, zygote formed start to develop into the embryo sac. In mammal, zygote formed begins to develop in the oviduct. In flowering plant, they form 3N nuclei and 2N zygote. In mammal, 2N zygote only formed. Part B. Give an account of the events which occur in oviduct leading to fertilization. Sperms get in contact with the secondary oocyte. The acrosome membrane of the sperm fuses with the egg. Next to its acrosome membrane ruptures, releasing the hydrolytic enzyme or digestive protease. Sperm head binds to the special receptor on the surface of the zona pellucida. Hydrolytic enzyme digests the path through the zona pellucida. One sperm penetrates into the cytoplasm of the secondary oocytes. Then the cortical granules, the lysosome, in the cytoplasm of the secondary oocytes release the enzyme which causes the zona pellucida to thicken and harden to form a fertilization membrane is formed, and the enzyme also destroys the sperm receptors on the surface membrane of the secondary oocytes. The secondary oocyte undergoes secondary meiotic division to form a haploid ovum and a polar body. The haploid nucleus of the sperm fuses with the haploid nucleus of the ovum. A diploid zygote is formed. Part C. What are the advantages of internal fertilization in mammals? Embryos are protected from hostile environment, predators, and mechanical damages. The embryos easily obtain nutrients, There is high chances for fertilization, reliable, and it is efficient. Reduced wastage of gametes. There is rapid growth due to optimum temperature and promotes parental care. Part C. What are the disadvantages of internal fertilization in mammals? Embryos are vulnerable to toxic chemicals produced by the mother. The fetus is entirely dependent on the mother. When the mother dies, the fetus also dies. The fetus may contract infections from the mother. The mothers are deprived of nutrients which the fetus obtains from them. Delayed fertilization since it relies on courtship. Long gestation period causes few offspring to be produced. Mothers may be exposed to predation during the period of pregnancy. (44) Explain the functions of the mammalian placenta as a barrier. Part B. Explain the series of events that will take place immediately after fertilization, which leads to the formation of the placenta. Part C. Describe how the negative feedback mechanism controls the menstrual cycle. Explain the functions of the mammalian placenta as a barrier. 1. Prevents maternal and fetal blood from mixing and stopping cases of agglutination occurs if blood is not compatible. It prevents some pathogens and their toxins from crossing the placenta. It protects the fetus against certain infections. It stops some maternal hormones and chemicals which are capable of causing damage to the fetus to pass from the maternal blood to the fetal blood. It keeps apart maternal and fetal blood, which operates at different pressures, and no harm is caused to the fetus. Part B. Explain the series of events that will take place immediately after fertilization, which lead to the formation of the placenta. Immediately after fertilization, the diploid zygote undergoes cleavage. Many small daughter cells form a solid mass of cells called morula. The morula reaches the uterus in 3 to 4 days. Mitotic cell division continues to form many small daughter cells, now known as blastomeres. The blastomeres arrange themselves into a fluid-filled ball cell called blastocyst. The outer layer of the blastocyst consists of the blastomeres called trophoblasts. While in the uterus, zona pellucida gradually disappears and the trophoblast makes contact with the cells of the endometrium of the uterus. Trophoblasts obtain nutrient from the endometrium of the uterus. Cells of the trophoblasts further divide mitotically and multiplies between 6 to 9 days. Trophoblasts is embedded within the endometrium, in which a process called implantation. Cells of the trophoblast differentiate into outermost membrane called the chorion and the innermost membrane known as the amnion. A sac-like outgrowth develops from the embryo gut and forms the third membrane called the allantois. Allantois grows outwards until it gets into contact with the chorion to form allantochorion, which develops into the placenta. Part C. Describe how negative feedback mechanism controls the menstrual cycle. High concentration of estrogen inhibits the secretion of follicle-stimulating hormone and luteinizing hormone by the anterior pituitary gland. This happens during the first half of the menstrual cycle, causing the level of follicle-stimulating hormone and luteinizing hormone to fall. However, when estrogen levels are very high, a surge of luteinizing hormone secretion occurs, which brings about ovulation. Towards the end of the cycle, estrogen and progesterone levels fall. Inhibition of follicle-stimulating hormone and luteinizing secretion are lifted. So the concentration of these hormones begin to increase, leading to the start of a new cycle. Thank you very much for taking part in reproduction in living organisms. Questions 41 to 45. Please do like, share, and subscribe.